So I would like to introduce the class for this semester. This is about multiscale modeling for data classification and algorithm design. Where I'm going to emphasize basically basic neuroscience and clinical ones. So the class will be made with my collaborator Thibault Lagache from Pasteur and Jürgen Reingober from INSERM. All right, so what are we going to study during this semester? Basically, the idea is to look into three aspects of the brain. One is modeling. The second is going to be simulations. And the third one is data analysis using classification methods to try to go into real-time prediction. So I'll spend now about 10 minutes to describe the general idea of why we're doing that, to present different questions we have, and then I'll introduce the syllabus of the class. So, as you all know, the brain is a multi-complex organ with multiple time and spatial scales as represented here. So you have here the spatial scale representing the integration from the nanometer, from protein that are so important, from membrane, from the guardian of the memory, that are located, for example, in neurons at synapses, and synapses, which are the contact between neurons, integrate the information. So at the cellular level, they can process the information, store the information, and then leads to local circuits, probably where some of the information is processed to have a higher degree of processing. And then everything is organized in brain regions. And finally, we have the whole brain. And the spatial integration is actually associated with the time scale going from, I would say, milliseconds for communications to seconds, which is the time scale usually of traveling of information from one region to another and processing. And then we go to hours, to memory, where I would say this engaged to minutes, to years, or tens of years, so that we can remember things. So how do we deal with this complexity? And I would say that in the last 20 years, neuroscience has moved in the different directions, from protein to subcellular organization, subcellular dynamics, such as calcium dynamics, to integrate everything into network level. So this is for the first type of model. We're discussing diffusion type model, electro-diffusion type model, to estimate, for example, the calcium time course or spike time course. And then the question is how things that happen at one scale, which is the scale of a single neuron, how does that integrate into multi-dimensional neuronal ensembles, which can involve hundreds, thousands, millions of neurons, or even more together. How this is integrated, and what is the meaning of integration? So this is still not understood, and we are thinking, and now I think the field is mature enough to say that this is part of neuroscience, to try to understand how this integration is made through the approach of computation, data processing, data analysis, and simulation, to see how these processes can be understood from the concept of the computer, the concept of calculations, and also the concept of how modeling and mathematics allow to better understand this organization. Okay, so what are we going to discuss? Mathematical multiscale models, how we go from one scale to another, from molecule to subcellular, as I explained previously. Numerical simulation is important to design and quantify, for example, the effect of the geometry. This is the example of dendritic spines. It is also very important, and I'll spend a class on this, on signal processing, when you have recordings from pipette or nanopipette, as we're going to see now. What is a signal? What can you extract? Don't expect machine learning or deep learning to find the features for you, because if nobody has seen any new features like this, don't expect the computer to find it for you. You have to know how to segment. You have an idea of what you're looking for, and for this you have to have a pre-idea of what the machine, or in terms here of the cells, what the cell is doing and what we can expect. And then it's okay. Once you have this, you can go to machine learning classification. After you segment your traces, you can classify and obtain a large amount of them. So statistical methods is still important to understand how, like in statistical physics, 100 years ago, the classical vision of Boltzmann going from molecules to thermodynamics was very insightful in terms of how we have understood how the physics go from one scale to another. And I think it's the same here in neuroscience, how and in cell biology in general. How do you go from one molecule to an ensemble of events that leads to a function? So this is the same approach that statistical methods can reveal, that allows, for example, for coarse graining. If you have, for example, 100 neurons that are doing almost the same, you can coarse grain all of this. But still, the ensemble can do more than one in particular. This is the case when you have synaptic depression that allows, for example, cells to behave synchronously, because as we're going to see, when the synapse is getting tired of sending signals, the fact that you have an ensemble sufficiently connected will synchronize the ensemble based on the synaptic delay or the synaptic fatigue. Okay, and then so statistical methods are important. We'll discuss some approaches like mean field model, how to average neuronal network from single neurons to manys, how to analyze ensemble of data. So this is quite important when you have statistics of, especially clinical statistics. How do you get some useful common behavior, common patterns into the behavior of the brain. So this year I'm not going to discuss much super resolution. Single particle trajectory will go more from single time series, EEG, brain responses, and if we have time, I'll discuss a bit of the methods we have developed for coma. So most of the time we have to do a reverse engineering problem, which is well expressed here. If you have many trajectories, for example, here are the blue trajectories, how do you reconstruct the underlying structure from many trajectories? So this involves usually stochastic processes, and to understand how do you account for stochastic behavior of molecules inside complex structure here, such as a network. Modeling synaptic transmission, we'll spend time on this, mapping voltage and deconvolution of voltage dye, because if you have a molecule that emit light and you have many of them, you have to understand how you convert this light into voltage so that you can quantify this. And usually, as we're going to see, we need electro-diffusion modeling. This is a model that accounts for, at the same time, diffusion and the interaction of molecules or ions due to an electric field that they are generating when moving. So we'll discuss also how we are going to model synaptic transmission from vesicular release to the activation of the postsynaptic part, where you have this blue molecule here that will affect the green receptor to generate a current. This is important also to quantify the geometry. Here, this is mostly the geometry of the dendritic spine, when you have a little head, which is like a ball or round, connected by a little neck, cylindrical neck to the main shaft. And since you have hundreds of thousands of these into a single spine, it will be interesting to understand how we can quantify this. I spend a little time here on discussing what is called the tripartite synapse, where you have interaction between neurons here and glial cells, leading to three compartments: the neurons, the extracellular space, or the space in between, and in purple, the glial cells. And I discussed, as I said a few minutes ago, if you have many neurons connected together, how can they generate a coherent response? You see here with a different color, each color is a neuron, and you see that when there is a spike here, then all neurons behave the same. This is due to their many recurrent connections. So one ensemble can create, let's say, as you see here, a long response of 2 seconds, which cannot be the response associated to one neuron. So altogether, they can reverberate and generate a longer response. Then we discuss a bit of how can we predict the brain using electroencephalogram. So you see you have your signal, and from the signal I will explain how you can generate the structure here, which is called a spectrogram, which is basically the connection as a function of time and the frequency. So how you connect the most important dominant frequency at a given moment of time. So for example, in here you see this is a frequency between 8 to 12 Hertz, where you see here this oscillation at 8 to 12 Hertz that is dominating here. So this is important because this can be used to evaluate, to quantify the different bands that are included or generated in the EEG in many conditions. It can be in when the patient is awake, during sleep, coma, anesthesia, or any kind of processor. And this is important, for example, for pilot. If you can measure the EEG, you can assess the degree of vigilance during action. So another important question we're going to discuss is how can we reconstruct networks such as neuronal network or glial network from calcium fluorescence imaging. And suppose here you can label cells so that when calcium is activated, you see this activated calcium. And so this is a movie of a few parts of the brain which shows few astrocytes here. So this is one part of the brain, and you can see the activation of calcium. This is the white here signal that sometimes is spreading, appearing, disappearing, and the question is what this is revealing about, first of all, the activity at a given moment of time, and second, about the underlying connectivity. In other words, if we see that there is, like we see here, a general correlations between the signal, can we infer that there is a specific type of connectivity in this ensemble of cells versus another ensemble which didn't show this kind of activity? And if yes, how can this be done? In other words, suppose you have an ensemble of astrocytes. We would like to use this possible time correlation across time to look at the possible path of activation. Let's say there is a first activated astrocyte that will activate most of the time a second one, then a third one, and a fourth one, and so on. This will reveal that there could be specific paths of action, specific path that underlie some cognition, some more elaborated process, and this is a way to, in some sense, reveal the way the information is processed in the brain, by saying that information is not necessarily just coded by spike, but by how spikes are delivered across an ensemble of cells, leading to also a spatial component into the way the information is processed. So we recently published an article, and I will discuss this, that is how we can first of all do some signal processing to extract and to extract information, which means we need to extract from this movie the region of interest, which are here represented by some region of interest where you have these astrocytes of different colors. And once you have these astrocytes with different colors, of course this is not that trivial, because you see sometimes the image will be heterogeneously represented. Some parts are more bright than other. So you have to use some kind of adaptive thresholding, which allows to extract the information about the astrocytes. And once you have the spatial organization, you can specifically look at the time dependency of the fluorescent in this region and do a general segmentation, which allows you to identify the peaks, which is a moment where you have here bright a spot of activation. And so once you have this, you can look at this in different regions and finally find out if there is a time correlation between this peak and that peak, or between this peak and a peak in a neighboring region. And from this, I will explain in the class how you can generate a path, a sequential path of activation. And when you sum this sequential path of activation, you can see which one of a given astrocyte and the next one in time was activated. And as you can see here, when doing this reconstruction, we found out that there are, for example, more systematic activation between this one and that one, which is here the higher yellow quantifying this connection threshold, compared to this red one, which is lower, suggesting that there is a preferential correlation between the astrocytes here that are in this graph, and they are connected by this yellow edge. All right, so now we'll continue by discussing this general question of when during anesthesia the patient is getting surgery, how do you monitor the brain? So this is what happened in the surgery room. An example, this is classical surgery, visceral surgery. And while the patient is getting the surgery, the brain is monitored using an array of electrode, and the classical monitoring of EEG is either Medtronic or Masimo, or no, there are other companies that have converted this EEG signal into a number between 0 and 100, here 36, and according to this number, between 40 and 60, the anesthesiologist is guided to place the patient in between this range to guarantee that the patient will be not too much and not too lower, getting to a smaller dose, or having a brain that is not enough anesthetized. So this allows, in some sense, to evaluate the brain, but is not predictive. In other words, you cannot use this to infer what would be the next concentration in the next few minutes. And we have also found out that actually the putting the patient between 40 and 60 is not necessarily what needs to be done, because a patient can be not enough or too deeply anesthetized in this band, and then this is not revealed by this index, but this is the subject of research. So this is how this interpretation is made. This is possible because over the last decades, people have understood how to read the EEG. For example, with frequency higher than 20 Hertz, which is basically the beta and the gamma band, this is associated to wakefulness. The beta band is the same. You can see here this event between 20 and 30. However, during stages such as in consciousness, you can see the appearance of two types of bands. One is the alpha band, which is between 8 and 12 Hertz, and the other one is the delta band, which is this very long and slow oscillations between around 0 to 4 Hertz. So this is the signature of what you would like to see during, for example, anesthesia. However, a high anesthesia, too deep anesthesia, is characterized by burst suppression, which are characterized by these events of suppression, here a flat line, which alternates with these fast oscillations, which is called the burst. And if you continue increasing the dose, in that case, you can even lead to isoelectric suppression, which is very bad in the strong way that minutes or more can damage the brain of this. And for example, when the brain is dead, it is shown here by a constant isoelectric state. All right, so I'll show you these examples where you can have here an EEG recorded in real time, and you see here the appearance of the two bands, the delta and the alpha band, that are dominating. And in the second video, you see what happened when you have this burst suppression, where there's a moment of flat EEG, and it is separated by this burst, showing that it happened quite often that patients are actually in that state. Okay, so how do you quantify the state of a patient? How do you quantify the brain activity and the propensity of a patient, for example, to be sensitive to anesthesia? In physical system, the way this is done is usually by looking at the response of a system going from one state to another. So for example, if the patient is awake and move to a state where it is dominated by this alpha and delta band, which is a different state, what is interesting is to estimate, for example, the transition from this state 1 to this state 2. And one way to do it is, for example, to assess when is the moment that you will lose, for example, one of the band, let's say the alpha band. And the first time can be a quantification of the level of sensitivity. Another parameter which also measure this transition duration is the state to go to from awake to a state where you will have the first isoelectric suppression, which I explained to you just a few slides ago, that this is associated with a deep sedation. So you can expect, for example, for patients that are very sensitive, that this time would be very short, so lambda will be very high. And for patients that are not sensitive, then this time will be very long, and thus alpha will be very small. So in that way, you can quantify, you can assess the degree of sensitivity to anesthesia by looking at this time scale, this number, these parameters that quantify the brain. Those are, of course, very interesting quantity because they depend on the entire brain or the age of the patients. So they are complex, but they can be simple in terms of measurements. Okay, so now let me describe the class. The first part of the class, part 1, will be associated to theory, and we'll discuss modeling and asymptotics for data analysis. I'll start with reminding what are the stochastic processes. So this should be background, Ornstein-Ulhembeck processes, and the Fokker-Planck equation, which is basically the probability density function for the stochastic processes. And we also discuss MFPT, which is mean first passage time. That is the time for a process to reach a certain region of the space when they start somewhere else. Then we will move on to discuss extreme statistics. That is, if you have 100 particles, what is the time for the first one to arrive to one place? And the redundancy principle is why in nature you have many copies of the same object in order to make sure that a function will be achieved. And I'll give the example of fertility, why you need so many sperms so that one will arrive to the right place at the right time. So to study rare events, what is interesting is to usually use asymptotics of the associated equation. The asymptotics will reveal, for example, how the time to arrive to one place for the fastest depends on the dynamics, let's say, for example, Brownian motion, depends on the geometry, depends on the number of particles, and so on. Then we'll discuss also an important part of the study of this quantity, which is how to make a simulation, and in particular when you have hundreds of thousands or millions or more of particles, how you can design hybrid simulations to avoid making a simulation in a space when it is not necessary. Then we'll discuss electro-diffusion and application. So this is basically how you model voltage in mitochondria, in dendritic spine, in protrusion of astrocytes, and so on. Then I want to spend a little time on spectral decomposition. This is what we said. What is a time frequency analysis? How do you estimate parameter from empirical data? We'll discuss a little bit of wavelet theory, especially for transporting a coefficient, which is important for artifact corrections. So this is part one of the class. This will occupy about 1 month. Another part of the class will be to discuss data modeling of cellular and neuronal network processes. We'll discuss mean field models of neuronal network based on short-term synaptic plasticity. Basically, this is a discussion of the general low-dimensional mean field equation, which is dynamical systems, based, for example, when you look at the voltage, the variable of depression, and facilitations, and how we can use the phase space to predict the behavior of a network. This could be, for example, excitatory network with depression type synapses, network with depression and facilitation, and how we can connect networks. So I'll discuss a bit of what happened when you can have non-Poissonian dynamics. Modeling neuronal networks, how can you add, for example, after hyperpolarization, this is a quantity that is not derived from synapses. This is voltage, electrical property of neurons. How can you include this in previous models? For example, to model Thalamo-cortical loops during anesthesia. I'll have a special class on modeling neuron-glia interaction because glia play a fundamental role in the regulation of potassium. If potassium is misregulated, you can go to epilepsy type behavior. So I'll discuss this. And if I have time, I'll discuss a little bit of metabolism. Then we'll discuss how to reconstruct graph and statistics representing this functional connectivity from calcium time series. This is what I presented at the beginning of this background introduction, and how we can represent neuronal and astrocyte networks through this graph theory. Then I'll discuss a little bit of brain development, how morphogenetic gradients are formed. This is important because those are the coordinates in the brain. And without coordinates, when you have a neuron that has to go from one place to another, it's impossible. So you need to have these gradients. I'll explain this. And I'll discuss a bit of how we can model brain wiring, specifically how axons are moving to find the target, which is the basis of neuronal migration. And yes, there is a combinatorics puzzle. I mean, if you have an axon in so many gradients, how these specific cells can go from one place to the destination place through a very labyrinth of difficulties. And still, this is how the brain is wired. So this is accomplished. This is so we try to see how, through this computational approach, we can understand the role of the gradient, what is the accuracy at which an axon or the tip of the axon will find its target. And finally, we'll discuss a bit of spatial statistics and image processing, and this is where we connect with deep learning. This will allow us to reveal how cells are organized during development, and we'll take the example of microglia, and we'll discuss what is the connection, what is the relationship between the neighboring cells, how cells are aggregated near, for example, a cell of a given type, and how we could use also classification algorithms such as dbScan to see how cells are clustered or not clustered and how the cluster evolve with time. And this allows, for example, to evaluate during development how population of cells such as glial cells or microglia or any other cells are evolving, which is still a question of interest. So finally, we'll discuss parameter estimation in real time. So real time is really, really challenging condition. And so how you can use EEG statistics in real time and with modeling to make prediction. So we'll discuss statistics contained in the EEG signal, different methods of decomposition of the EEG, and how we can decompose the brain, not exactly with Markov chain, but with equivalent, which is called a state chart decomposition. We'll discuss a bit of empirical mode decomposition and power spectrum analysis to extract specific features inside a band, which is called the alpha band. Designing predictive algorithm for general anesthesia. So how do you design, in general, real-time algorithm? This could be for anesthesia, this could be for sleep, this could be for any action you would like to detect, which is probably an important aspect for future applications when new electrodes will be positioned in airports. So we will be able to monitor the brain and act on the brain using this new type of information, this new signal. So I'll discuss this. Classification algorithm, that's also very important because you want to make sure that you're in a given state. So for example, can we predict the outcome of coma? Is the person going to survive or not? If the person is surviving, with which accuracy? How to differentiate Parkinson versus essential tremor. So we also will discuss this part if we have time. And the class is the evaluation, about 40 hours project, which the result should be The result is I need a 2 pages report where each participant write a title, put his name, a little abstract, a little background, a result section, a little discussion, and 1 or 2 reference. And then there's a 20 minutes oral presentation on the PowerPoint, followed by 10 minutes of questions. Okay, I wanted to illustrate some of what we're going to study, but I think it's not necessary. I think I'll stop here, and I welcome everybody to the class. Thank you.